What did Diplodocus look like? Not just its bones, but its skin pattern. A new study on fossil skin from juvenile Diplodocus proposes the first evidence of sauropod color patterning using microscopic structures linked to melanin.
We discovered that the Diplodocus skin was preserved three-dimensionally, fossilizing the cellular detail, including the original color pattern.
The skin comes from the Mother's Day site in Montana's Morrison Formation, a Jurassic deposit now excavated by elevation science. To understand this, you need one word: melanosomes. Tiny structures inside cells that hold melanin, and in living animals, their shape and arrangement can contribute to color and patterning. In fossils, melanosomes can sometimes be preserved as molds or impressions, basically footprints left behind in the tissue. The team used scanning electron microscopy and elemental mapping to see what the skin was made of at a microscopic level.
This discovery was made possible with a machine called a scanning electron microscope. It's essentially a very, very powerful microscope that allows you to see cellular detail at amazing quality.
They found the outermost skin layer, the stratum corneum, preserved three-dimensionally as clay minerals, aluminosilicates, with flaky, sheet-like textures. Within and around that outer layer, they describe two distinct microbody shapes associated with carbon-rich signals.
One were the oblong type, which are pretty usual in what you expect to see in terms of melanosomes, but the other was disc-shaped and flat.
Crucially, these structures can appear clustered together rather than spread evenly, which the authors interpret as evidence for some kind of speckling, which is a really neat hint that any pigment-related patterning may have been patchy or speckled rather than one solid color.
Now, the reason why this was so surprising to us is because the closest modern animals that we see today with flat melanosomes are what you see in colorful bird feathers. Now, of course, this doesn't mean that Diplodocus was vibrantly colored, but it is certainly bizarre that these animals had melanosomes like this.
But here's what this does not give us: the exact color of Diplodocus. What it can put on the table are melanin-based colors, the family of tones melanin commonly produces in living animals, like blacks, browns, reddish browns, but not a particular shade or palette. The paper emphasizes that patterning signals can survive even when exact colors don't. But the implication is still big. If these microstructures are truly melanosome related and they're preserved in place, that means sauropod skin can preserve more about appearance than we previously thought, opening doors to testing patterns in other skin fossils too. This study suggests they had the biological tools for more complex appearance than we used to assume. And now we have a method to test that in fossil skin.